So let's move on to the functions. So if we are matching the structures to the functions right now, just a disclaimer, don't look at your handout while you're doing this because I forgot to mix them up. So the structure on the left in your handout is there, and now you have the answer to the function, but it will make it more easy for you to write in your handout the actual function. But don't look at it while you're doing this. So looking at the structure-function relationship, I want you to pause again and take a little bit of time to go through and move all of these answers into the structure part. You will see that I couldn't fit all of the structures and functions on one page. So not all of the functions are here for every answer. You just need to go to the second page and start where the last page ended to finish it. So take a pause and we'll meet back. So hopefully you had a little bit of time and you had some success. Like I said, I don't care if you get one right or if you get all of them right. I just want you to see where you are before we've actually talked about it. So based on the reading, based on any previous information that you have known, I want you to get an idea where you are. And then that way, if you already can do it, then you're great with this objective and you can move on. But if you only got a few, you know that you need to work on a little bit. So coming to the first one, the structure that conducts the action potential, that is the axon. We will get into the specifics of all that in one of the upcoming chapters. The part that receives the synaptic input, we kind of talked about it when we focused on the synapse, but the part that mainly receives the synaptic input is the dendrite. There can be other synapses on the cell body and also on the axon, but just for basics, most of the reception is on the dendrite. The part of it that contains the genetic information is in the nucleus. Cellular respiration provides energy in the form of ATP. Another way to say this is it's the powerhouse of the cell, but a lot of students just memorize that term and they don't actually recognize that it relates to using oxygen in order to fuel the breakdown of glucose and other molecules so that then you can create ATP, right? Powerhouse of the cell is an easy thing to remember. The actual process of cellular respiration in the mitochondria is a lot more difficult. The structure that is going to synthesize membrane-bound or secreted proteins, that is the rough ER. ER just stands for endoplasmic reticulum. I don't have smooth ER listed on here, but the smooth ER is just the lipid synthesis. But what, for our purposes, really what we need to focus on are those membrane-bound proteins because we're going to be talking about a lot of them. And then just in general protein synthesis, that are the ribosomes. So the ribosomes make the proteins. The rough ER makes proteins too because ribosomes are embedded in the rough ER. That's why it looks rough, because it's got the ribosomes and their little dots. Then the membrane-bound and free ribosome collections, which means if they're ribosomes, then we're talking about protein synthesis. Those are the Nissl bodies, which are specific to neurons. The structure that leads to cell structure and transport, part of the cytoskeleton, those are the microtubules. And then another part of the cytoskeleton that is involved in structure, transport, and then cell motility because it's at the cell membrane and then it changes in response to things and then it can push or pull the membrane. Those are the microfilaments, and you will hear me refer to them as actin in the future. So whenever I talk about actin, this is what we're talking about. The connection between two neurons, that is at the synapse, like we saw in the image before. The part that contains neurotransmitters in the presynaptic membrane, we also talked about that. Those are the synaptic vesicles. And then at the end of the axon, The part that releases the neurotransmitters, that is the presynaptic membrane. So that's this section. Now I'll go and work on the last half of it. Let's see, so we were at the end of the axon, thing that releases neurotransmitters. That was the presynaptic membrane, because the axon is the thing that is transmitting the information. So it's before the synapse. And then on the receiving cell, the part of the membrane that receives the neurotransmitter signal because it contains the receptors, that's the postsynaptic membrane. And then the thing that is released from the presynaptic cell that is going to communicate to the postsynaptic cell, this is actually a typo. I don't know why I have contains receptors. I think I was just looking at this. Sorry, that's a typo. Cross that out. It doesn't belong there. I was like, wait a minute, what's that? Okay. So it does not contain receptors. So this is the thing that's released from the presynaptic to communicate to the postsynaptic cell. That's the signaling molecule, right? And the signaling molecule are the neurotransmitters. They don't have receptors. Anyway, so then the thing that binds the neurotransmitter to change the receiving cell or to transduce the signal into the receiving cell, that is the receptor. It receives the signal, so it recepts the signal. And then the structure that increases the speed of an action potential is the myelin, and we'll look at two different types of myelin in our next exercise, or one of our next exercises. And then the spaces that are in between the myelin, you saw in the picture before, those are called nodes of Ranvier. So Ranvier is just the person who discovered those. And then, dang it, I have a lot of typos on here. I'm really sorry. So this is supposed to be synthesizes ribosomes. Anyway, that is the nucleolus. So the nucleolus synthesizes ribosomes. And then the place on the cell where all of the synaptic input adds up. So summation of synaptic inputs that are going to trigger or not trigger an action potential. And that is at the axon hillock. So that's that space where the cell body meets the axon. So again, I encourage you to pause the video, go back to your own copy of the Jamboard, and work on this a little bit more until you've got it down.